PharmaPal.
You're listening to Bloomscast, the official podcast of Blooms.io, home of the bioactive library for mass spectrometry instant optimization. Let's dive into the latest in natural chemistry, bioactive discovery, and the future of lab innovation.
So, our mission today, basically, figure out what network pharmacology actually is. How did we get here?
Yeah.
And why is it suddenly so important for understanding things like traditional Chinese medicine or TCM and, natural compounds in general? We want those aha moments, but keep it digestible. So, where did the whole hunt for medicines even start? Way back.
Well, the very earliest methods, it was pretty much looking around, exploring nature plants, minerals, based on knowledge passed down. Think medicine men, ancient healers, priests, their materia medica. It was all very observational, trial and error.
Right, very hands-on, experience-led, and then things get a bit more systematic.
Exactly. That led into what we call classical pharmacology. Scientists started testing specific small molecules, seeing what they did to cells or even whole animals, a more controlled analytical approach.
And then the big one, sequencing the human genome. That must have shifted things dramatically.
Oh, absolutely. Total game changer. That really kicked off the era of target-based drug discovery. Some call it reverse pharmacology. The thinking became, okay, find a specific protein involved in a disease that's your target, and then screen loads of chemicals to find one that messes with it.
One drug, one target, one disease idea.
That's the one. Very focused, very reductionist, really. You'd screen these huge libraries of chemicals against just one protein.
Like searching for a single key, for a single lock, a magic bullet. And tech helped refine this.
Massively. The IT revolution, all the omics, genomics, proteomics, plus huge leaps in computing power. Suddenly you could predict things computationally, like, is this molecule likely to even be a drug? Drug likeness. How might it bind to the protein? We could even model its AD-MET properties.
That even met.
Yeah. Absorption, distribution, metabolism, excretion, toxicity, basically how the body handles the drug, all predicted by computers. So it moved away from that holistic view of traditional medicine towards these very specific targets.
Sounds super efficient. But there must have been downsides. Why did we move away from the magic bullet?
Well, the big limitation was complexity. Most diseases, especially chronic ones, aren't caused by just one faulty protein. They're multifactorial. So a drug hitting only one target, it might cause side effects because that target does other things too. Or it just wouldn't be very effective against the whole disease network. You're only tackling one small piece of a complex puzzle.
That makes a lot of sense. And that realization kind of brought us back to looking at those original sources, natural products.
Exactly right. There's been a real renaissance for natural products. Why? Well, they have incredible chemical diversity. Many have been used safely for ages. And crucially, we now understand they often don't just hit one target. They're naturally multi-target. Actually, one of the sources mentioned, over 100 natural product-based drug leads are in clinical development right now.
Wow.
Yeah. The understanding now is that most diseases involve multiple dysfunctional proteins, so hitting multiple points might be better.
So we needed a better way to understand, and maybe even design, these multi-target effects. And that's where network pharmacology steps in.
You got it. Network pharmacology, or NP. It's a newer discipline focused on understanding drug actions precisely through their interactions with multiple targets. It uses computational power to map out all these molecular connections within a cell, like a complex web. This is super useful for understanding, say, how a traditional herbal formula, with all its different compounds, actually works in the body.
Okay, so it's not just one lock, one key. It's like a whole system of locks. And seeing how a set of keys interacts with everything. What can we actually do with NP?
The applications are pretty broad, actually.
Yeah.
It helps us decode the complex relationships between botanical formulas and the body. It can point us towards new drug leads, new targets we didn't know about, and even help repurpose existing drugs find new uses for old medicines by looking at their network effects in an unbiased way.
And you mentioned traditional knowledge can guide this.
Yeah, that's a really interesting point. Traditional knowledge can give us clues about which targets or types of chemical structures might be worth investigating with NP tools. It's a way to bridge the old and the new.
Okay, this is where it gets really cool. Combining NP with systems biology. That sounds powerful.
It really is. Systems biology gives us the big picture of how everything in a biological system, genes, proteins, metabolites, fits together and interacts. Combine that with NP, and you can start to rationally design what are sometimes called promiscuous drugs.
Promiscuous.
Yeah, sounds a bit weird, but it just means drugs designed intentionally to hit multiple targets in a controlled way. The potential payoff is huge. New therapeutic options, maybe safer and more effective medicines.
So NP didn't just spring up overnight. It built on earlier work, like network biology.
Right. Network biology came first. It's based on the idea that cellular processes aren't isolated, they're interconnected networks. And there might be universal laws governing these networks. It gave us a new framework for thinking about biology and disease in the 21st century. Early on, people developed computational approaches, things like analyzing scientific literature.
Like LMMA? What was that?
LMMA, literature mining, and microarray analysis. It was an early technique. You'd combine info pulled from scientific papers with data from microarray experiments. Those measure the activity of thousands of genes at once. It helped researchers piece together gene networks, make sense of massive data sets. It was useful for building specific networks, understanding pathways.
So we started seeing these complex webs, and that revealed something crucial about how drugs work, this promiscuity thing.
Exactly. As researchers mapped out drug interactions, they found many drugs weren't as selective as they thought. They could bind to multiple targets, that's the promiscuity part. And, the flip side, a single target could interact with lots of different molecules. That's polypharmacology.
So one key might open several locks, and one lock might accept several keys.
Pretty much. It's a good analogy.
And people started building these drug target networks. What do they show?
Well, they used databases like DrugBank, which lists drugs and targets, and OMIM, which is about genes and genetic disorders. They expect to see neat, isolated lines based on the one drug-own target model. But instead, they found this dense, interconnected network, lots of polypharmacology, drugs hitting multiple things. They also noticed loads of follow-on drugs, new drugs targeting proteins we already knew well, kind of highlighted the limits of just focusing on single known targets.
And this polypharmacology,
Yeah.
hitting multiple targets, that wasn't always seen as good, right?
No, initially it was often viewed as a negative, potential for side effects. The goal was often more selectivity. But as we understood biology better, especially for complex diseases, the thinking started to shift. Maybe hitting multiple targets at once is actually beneficial. Maybe it's necessary for effective treatment.
Which brings us back to network pharmacology. It was Hopkins who really crystallized this idea.
Yes, Andrew Hopkins was key. He really pulled together network biology and polypharmacology. He coined the term network pharmacology and argued it was the next paradigm in drug discovery. He saw it as the way forward for multi-gene diseases, complex conditions, even towards personalized medicine, what he called e-therapeutics.
Did he suggest ways to actually create these multi-target therapies?
He did. He outlined 3 main strategies. 1, using cocktails of existing single-target drugs. But he pointed out issues like patients actually taking all the pills and drugs interacting badly with each other.
Okay. Strategy 2.
Multi-component formulations, like traditional remedies, basically. But concerns there were about metabolism, bioavailability, getting the stuff into the body, and safety testing.
And the third one.
Designing single compounds that have selective polypharmacology. So they hit multiple specific targets, but in a planned, beneficial way. Hopkins thought this was the most promising. Easier dosing, potentially smoother regulatory path. Metformin, the diabetes drug, is often cited as an example. It seems to hit several targets.
So network pharmacology isn't just about finding new drugs then. It has wider uses.
Definitely. It's being used to develop tools like Cypher and CypherHit. These use protein interaction network data and disease characteristics, the phenotypes, to predict disease-causing genes. They can confirm known disease genes, but also flag new potential candidates.
That's amazing, using networks to uncover the genetic roots of disease.
Exactly. You can explore these phenotype-genotype networks to find genes related to specific conditions, see how genes cooperate. There's also network-based gene clustering grouping genes that behave similarly under certain conditions to understand physiological responses, and developing miRNA-based biomarkers. miRNAs are small molecules that regulate genes. NP helps build networks to find useful disease markers, even creating these complex drug-gene disease comodules to see how multiple genes, drugs, and diseases are linked, revealing potential new drug uses.
This sounds incredibly powerful, and it feels particularly relevant for traditional medicine, doesn't it?
It really does. There's this growing appreciation for natural products. They've been sort of optimized by evolution, right? And researchers are using computational methods to try and understand the logic behind traditional systems like TCM. There are huge databases now documenting herbal recipes, ingredients, molecules, especially for things like cancer treatment in TCM.
Which leads to network ethnopharmacology. What's the ethno part add?
Network ethnopharmacology, or NEP, is just applying NK specifically to understand traditional medicines and botanicals. We talk about ethnopharmacological networks. A key figure here is Shaoli. Lane proposed using these networks to explain concepts like ZHEN in TCM, which is sort of like a syndrome or pattern of disharmony, and the multi-target way TCM formulas work.
Trying to find a molecular basis for something like ZHEE. That sounds ambitious.
Hugely ambitious. Laya's group tried using a neuroendocrine immune NEI network model. The idea was maybe TCM concepts like hot and cold ZHeng map onto things like hormones for HT and immune factors for cold, linked by neurotransmitters. They built this complex NEI network using data from pathways, metabolism, gene regulation. Their work suggested cold's ZHN was strongly linked to energy metabolism and genes involved in neurotransmitter, hormone, and immune signaling.
Wow. Bridging ancient concepts and modern molecular biology. Are there specific TCM databases for this?
Yes, things like TCM Genodite. They collect info on TCMs, related genes, diseases, effects, ingredients, all pulled from scientific papers. It's a massive resource. People have also built herb networks to understand why certain herbs are combined in TCM formulas. They've developed models like DM to find the rules of combination, and even tested predictions experimentally, like looking at effects on blood vessel growth and angiogenesis.
So it helps make developing therapies based on traditional knowledge more, rational.
That's the goal. There's this network target-based paradigm, NIMS, proposed to understand synergy and combinations. Algorithms like NIMS aim to help design multi-component drugs based on that traditional wisdom. We're also seeing studies combining experimental work with gene expression data, like the Sai Wu-Tang example, to probe mechanisms. NEP is increasingly used to study specific formulas like Kingfei Shian for inflammation, with experiments then confirming the network findings.
And building the right databases for natural products is key to all this.
Absolutely crucial. The first step in NEP is usually digging into databases. You need info on the bioactives databases like UMPD, TCMSP, Supernatural hold molecular structures and chemical data. Then you predict targets using databases like BindingDB or Chembol, often based on chemical similarity to known drugs. And these link out to other resources like Uniprot for protein info, KEGI for pathways, OMIM for diseases. You build up layers of information. Integrating multiple databases gives a richer picture than relying on just one.
Okay, so you gather all this data. How do you actually build a network visually?
Well, a network is just a map. You have nodes. These could be bioactives, targets, diseases, tissues, and edges connecting them, representing the relationships, like binds to or is associated with. You can build them bottom up from experimental data or top down from existing knowledge. Often it's a mix. The key is making sure the nodes and connections actually reflect biological reality.
And you need tools to see these complex maps, right?
Definitely. Cytoscape is the big one. It's open source, widely used for visualizing molecular networks. You can integrate different data types, and there are loads of plugins, apps for specific analyses or linking to databases. There are others too, like VMP, Connectivity Map, GIFI, depending on what exactly you want to visualize or analyze.
We've talked a lot about PCM. What about other traditional systems like Ayurveda from India?
Good question. Ayurveda also has a hugely sophisticated system with complex formulations used for millennia. There's the Traditional Knowledge Digital Library, TKDL, which documents a vast number of these. But interestingly, specific network pharmacology research on Ayurveda was relatively limited until more recently. Our own group actually used a very famous Ayurvedic formula, Triphala, as a kind of case study to show how NEP could be applied.
Triphala. I think I've heard of that one. What is it, and why use it as a model?
Triphala means 3 fruits. It's a blend of Amalaki, Emblica officinalis, Vibhitaka, Terminalia balearica, and Haritaki, Terminalia chibula. It's incredibly widely used in Ayurveda for digestion, oral health, skin, even as supportive care in cancer. It's seen as a general rejuvenator, good for eyes, heart, slowing aging. It's packed with polyphenols, which likely explains its known antimicrobial properties. And because it's in so many Ayurvedic formulas and has such broad reported benefits, it was a perfect candidate to see what NP could reveal. We wanted to map its effects across the human proteome, the diseasome, disease-related genus proteins, and even microbial targets.
So what did the NP analysis show? What about the bioactives and their human targets?
Well, we found a good number of known bioactives in each fruit. Combined, it's chemically quite rich. We focused on what we called Score 1 bioactives, those with higher confidence predictions for hitting targets based on database searches. Interestingly, several were common to all three fruits, like scopolamine, ellagic acid, tannic acid. Looking at human protein interactions, we found 36 of these bioactives potentially interacting with 60 different human proteins, over 100 interactions in total. Some compounds like quercetin and ellagic acid seemed particularly multi-targeting. The targets themselves were diverse enzymes, coagulation factors, DNA repair proteins, inflammatory mediators, suggest triphala hits multiple physiological pathways.
And these targets were linked to specific diseases.
Yes. Those 60 targets mapped onto 24 broad disease categories covering 130 specific diseases. The big ones were cancers, cardiovascular diseases, nervous system disorders, and metabolic diseases. For instance, links to 40 cancer types via 25 bioactives and 27 targets. Cardiovascular links involve 12 bioactives and 11 targets across 18 conditions. Similar patterns for neurological issues like Alzheimer's, Parkinson's, and metabolic problems like diabetes and obesity, plus links to autoimmune conditions, colitis, gout, osteoarthritis. The list goes on. It really paints a picture of triphala, potentially modulating core processes like inflammation, cell growth, metabolism pathways relevant to many different conditions.
You also looked at microbial targets. What did that network show?
Right. Triphala is known traditionally for gut health and has demonstrated antimicrobial effects in labs. Our network analysis found 24 of those Score 1 bioactives potentially targeting proteins in 22 different microbes. These included targets linked to diseases like TB, malaria, HIV, hepatitis C, candidiasis, even SARS. We saw potential interactions with key microbial proteins, for example, hitting an enzyme essential for the TB bacterium's cell wall, a target not present in humans, or potential inhibition of HIV reverse transcriptase, or proteins needed by the malaria parasite or HPV. Compounds like epigallocatechin, 3-gallet, from green tea, also in triphala, and quercetin showed broad antimicrobial targeting. It really backs up the traditional use and suggests mechanisms even for drug-resistant bugs.
So pulling this all together, NP looks like a really versatile toolkit.
It absolutely is. It's gaining ground as a novel, potentially more cost-effective and predictable approach to drug discovery. Like the source table summarized, it has huge implications for traditional medicine, providing evidence, understanding mechanisms, ensuring safety, designing new network-based therapies, and in mainstream pharmacology, finding leads, understanding side effects, repurposing drugs, plus fundamental drug research, finding novel targets, understanding disease pathways, designing experiments, tackling complex diseases.
Okay, but it can't be perfect. What are the limitations, the catches? Especially for traditional medicine.
No, you're right. There are challenges. A big one is relying on databases. They can have gaps or discrepancies. Data curation is key. Also, how traditional medicines are prepared, boiling, fermenting, can change the chemicals. What's in the database might not be exactly what's in the final dose.
So how do you fix that?
Well, one solution is using modern chemistry techniques like UPLC MSMS to directly identify the compounds in the actual formulation being studied. Makes the MT analysis much more accurate. Another challenge is the role of vehicles in traditional medicine, things like water, milk, honey used in preparation. They can affect absorption and metabolism. We need more research on their effects.
What else?
Access to comprehensive target databases can be an issue if you only rely on free resources. You might miss things. So integrating multiple databases is better, and crucially, experimental validation of predicted targets is really important. You need lab work to confirm the computer predictions. And finally, synergy is complex. These formulations have many compounds hitting many targets, some activating, some inhibiting.
Right, simple models might miss that complexity.
Exactly. In vitro or in silico tools can't fully capture what happens in a living system. So NP is best seen as a powerful first step. It gives you a map to guide the essential in vivo experiments and clinical trials, hopefully saving time and cost. And using omics technologies based on network predictions can help unravel those complex synergistic effects.
This has been incredibly insightful. Network pharmacology really does feel like a bridge between ancient knowledge and very modern science.
It absolutely is. It offers a huge potential to systematically explore traditional wisdom and tackle modern drug discovery hurdles, finding new leads, repurposing old drugs, designing rational combination therapies. It's a powerful lens. It's kind of a reverse approach, using modern tech to deduce the molecular logic behind formulations based on their known components. Of course, the field's still evolving, data is constantly updated, but even with limitations, it's providing valuable insights into knowledge that's been hidden in plain sight for centuries.
So, a final thought for you to chew on. As we get better at this, how might blending network pharmacology with artificial intelligence truly revolutionize things? Could AI analyze these complex networks and traditional knowledge bases to discover novel therapeutics, perhaps even personalized ones, much faster than we ever thought possible? Definitely something to think about.
PharmaPal.